To explain it really simply, we shine microwave energy at gas molecules, and we kind of listen to how they spin. And that's how we are able to measure with very high accuracy and precision the kind of fingerprint or voice print of that chemical compound or the molecules we're looking at.
That's Damien Fischer, the Chief Commercial Officer of BrightSpec, a pioneer tools company for molecular analysis. Their molecular rotational resonance technology brings unmatched clarity to small molecule characterization. With tools like nano-MMR, iso-MMR, and spectra-MMR, BrightSpec is helping labs in pharma, chemicals, and consumer goods get answers they couldn't before. Welcome to the Biotech Stack, where we explore the cutting-edge technology fueling the future of life sciences and biotech. From next-generation lab automation and AI-driven drug discovery to breakthrough biomanufacturing and research tools, we dive deep into the innovations transforming health, food, energy, and the businesses behind them. I'm Corinne Regan, your host. And in today's episode of the Biotech Stack, I'm joined by my old friend and previous sales leader, Damien Fischer, the commercial officer at BrightSpec. Damien shares his journey going from lab bench to leading the commercial efforts for research technologies. We dive into how BrightSpec's technology is shaping everything from drug development to consumer products, giving scientists faster, more reliable answers, and unlocking discoveries that were once out of reach. Let's get started. Hi, Damien. Thanks for joining us today. I'd like to jump right in and learn a little bit more about yourself, your background, and potentially a little bit more about your current role at BrightSpec.
Well, I'm an analytical chemist by training. I got my degree from UC Santa Barbara a long time ago, and I started out in the laboratory on the other side of the bench working on analytical instruments at a couple of chemistry companies, a consumer packaged goods company, and then a diagnostic company. And I got recruited to Agilent pretty young. And I jumped across to the vendor side as a service engineer and application scientist. And then I was quickly recruited in the early stages of my career at Agilent to the dark side. And I joined the sales team about 20 years ago. And then I kind of took on every commercial role at Agilent, from product specialist and account manager roles to business development to sales leadership roles. And at the end of my career at Agilent, I got into mergers and acquisitions, and I was on a corporate team, and I moved up to Santa Clara, where I kind of fell in love with the idea of joining a smaller startup business, which was how I ended up leaving Agilent, and I joined a company called SIF Technologies, based out of Christchurch, New Zealand, as their global VP of sales and marketing. That went really well. I had a lot of fun. We kind of did a 0 to 1 launch of a new product. I started a team in the United States and North America, and then we grew to the size where we decided to jump into building a European team. I managed that team as well. And right kind of when we were in a transitional point, I got recruited by BrightSpec to be their chief commercial officer, and that's where I've been for the last year.
Can you share a little bit more about this commercial role at BrightSpec?
Absolutely. So about a year ago, I was contacted by a search committee for this company, and they were getting ready to do a full commercial launch of a brand new platform of instruments using a new technology called MRR, which stands for molecular rotational resonance. And at that point, they were looking for somebody who had expertise at a big company on the commercial side, but also in the startup launching a new product. And it just was kind of a clincher that I also had a lot of experience in the same markets in small molecule volatile analysis, GCMS and LCMS, which really aligned with the objectives of the company.
Okay, great. So speaking of the technology at BrightSpec, my understanding is it's a fairly new technology on the market, but not a new technology in an application standpoint. Can you kind of dive into that a little bit more? What does BrightSpec do, and what is that technology?
Absolutely. So I'll start with what is the technology. So MRR, molecular rotational resonance, to explain it really simply, we shine microwave energy at gas molecules, and we kind of listen to how they spin. And that's how we are able to measure with very high accuracy and precision the kind of fingerprint or voice print of that chemical compound or molecule, or the molecules we're looking at. So every molecule spins a little bit differently. And we can tell exactly how they spin in three-dimensional space, giving us really well-defined structural identification.
What industry is this important in? When would you need to know the structural identification based on its spin in the world of maybe pharma, or what industries that you guys are part of?
So I'm as a kind of a new technology, and the way that we are able to measure molecules, the key is that they have to be in the gas phase. And so we need to be able to take the sample and the matrix from a customer, and then we need to be able to introduce it into the instrument. Right now, our target markets are pharma, consumer goods, flavors and fragrance, and academia, so research and development. So we kind of cover the entire gamut of analytical testing.
Gotcha. And is this just a capital equipment, or is there a software technology that exists with this platform as well?
So this is a full solution. So we actually have kind of three unique interactions with our solution. Number one, the measurements that we take are dependent upon quantum chemistry models. And so we actually have a software that allows our users to be able to actually simulate their analyses beforehand virtually. And so you can actually log into our software. The initial interaction would be with one form of our software called BrightHub. You would query the compounds of interest that you have, and it'll give you some information about your targets, whether they've been run on an MRR platform before, and give you some of the specific structural information about that. If we have run it before, it'll actually give you a theoretical spectra, as well as measured spectra if we've done it. If it hasn't been run before, then you have an option to actually simulate that molecule of interest, and then it'll give you a theoretical spectra after we run a quantum chemistry simulation. So that's kind of that front-end interaction. And then we have a fully automated hardware platform that allows you to load your sample. We have auto samplers and some automated sample preparation, and run it through the instrument, get the detection. And then on the reporting side, on the back end, we have a data analysis platform as well. So it is kind of that combination of software, hardware, and application to give you the full solution.
Does this technology exist elsewhere? Are there maybe some competitors in the space? And if so, does there have some type of special sauce to the BrightSpec technology and software that makes you a little different in the industry?
Absolutely. So I would go back to the original question, where is MRR from? So it's actually a theoretical capability, a measurement technique that was invented about 60 years ago. And we kind of have benefited from Moore's law. So as we have gotten more and more capability on computer capability and power, as well as with electronics and hardware, we've been able to actually take something that was about the size of an entire laboratory and required a supercomputer to be calculating the results off of one measurement, which would take years to do. And this was one measurement was a PhD thesis when this technology was invented. And originally, Agilent, or HP, attempted to commercialize MRR 50 years ago. It didn't quite have the capability necessary to unlock commercial interest back then. Fast forward to 10 years ago when BrightSpec was founded, we could now actually create an instrument that was about the size of a small refrigerator and was able to give you data in minutes, if not hours, rather than days or weeks. And so, that was kind of the origination of MRR as a technique. What's our special sauce? The uniqueness is that MRR gives you direct, unambiguous structure, and you don't have to ionize. There's no fragmentation, and limited to no separation or sample prep. So we're not guessing as to what the compound or molecule is. We actually are giving you that structural information with high precision as a part of our measurement. Along with that, as we mentioned, we have that ability to do that virtual simulation, which is actually quite valuable. It speeds up analysis, lets you know if actually buying standards or references and setting up your full measurement is necessary so you don't waste time and resources. And we solve problems that other instrumentation can't. When you look at specific structural isomers and structurally similar compounds, and even chiral compounds that are extremely difficult to analyze on other traditional platforms, we do really well. We also don't require chromophores. And so there are some explicit applications and target molecules that are very difficult to do on a traditional GC or LC that just happen to resonate really well on the MRR platform.
Because this provides such unambiguous confirmation, is this something that will take place of what already exists in the workflow when you're characterizing molecules? Or is this something that will be complementary to? Or can it be both?
Well, as of right now, we really are looking at targeting orthogonality. So where there are additional insights necessary and customers or prospects are interested in trying out a new technology, MRR is a partner in the application. We are not positioning ourselves as a direct competitor or a replacement technology.
How do you position BrightSpec? You are a capital equipment company. There's obviously software and it's a full solution. But sometimes those capital equipment sales can be a little bit more complicated to get into. Large pharma, even small pharma, definitely. How do we overcome that? What is the messaging behind this long-term asset?
Absolutely. So right now, our primary goal is to focus on critical problems for our prospects. This is not a nice to have technology. We want to gear towards solving problems that can't be solved by another platform. How do we do that? We're able to shorten workflows, eliminate ambiguity like we just talked about with that structural identification and clarity. And we can kind of reduce the overhead with the ability to do that virtual modeling and to automate your workflow. In many instances, we are going to be faster, less technical, and we can even be brought out of the traditional laboratory because of how simple it is to interface with the instrument. And then you go back to my team. We really guide the customer through the entire process. Because this is a new technology, we're very invested in owning the solution as a partner with our customers. So we'll guide it through method development and the application as well. And we're not just selling the platform and giving it over to a customer to figure it out on their own. We're going to be there lockstep the entire way.
Going a little bit bigger picture here, I wanted to talk to you about some of the macroeconomic changes we've experienced here in the U.S. over the last year. We're all aware, the NIH has lost some funding. We've had tariff issues. Curious, even at this stage in BrightSpec's journey, has these impacts affected your outcome maybe this year or into the next year or so?
Well, I would be insincere to say that it hasn't made an impact at all. It's a pretty tough time right now to be a startup company launching a brand new product in the economy that we have. But that all being said, it also leads to more criticality in decision-making by our prospects. And we believe that we are very well differentiated. And so if you look at those tighter budgets, what's important? Confidence in your data has never been more important. We give that level of precision and we reduce the ambiguity around your results. People are looking for workflow consolidation and orthogonal validation. So you have that confidence. And they want to reduce the risk that they have in their products, in their laboratory, quality control. We offer all of these things. We're also faster in many instances, and we have lower overhead, and we're a green solution. We don't require solvents. We don't require harsh chemicals along with the workflow. And so you package that all together, I think we're in a really good space.
I love that answer. One, it's a positive spin on what's happening in our economy, which it is a tough economy, especially in this marketplace. And so to be able to provide all those, the data integrity is the most important part. And to be able to find a solution that even in this market still makes sense, I think is wonderful. With that said, I know we are sort of still a brand new technology. However, are there additional innovations that your team is looking at over the next 3 to 5 years?
Well, I wouldn't, I don't want to be too on the nose, but we do push for kind of a relentless innovation. We know as a new technology, we need to found ourselves in a space where our customers are confident, but we also need to continue to add capabilities to the platform. One big area is just about sensitivity and continuing to work towards being able to get lower and lower in our detection. And that's going to be something that we work on for years to come. Another thing is our software, making it easier to use, improving the user interface, the confidence in the results is all very important. And there's always more that we can do to establish a more powerful software. But one of the biggest unlocks for us as a new technology is integration. We are a hyphenated detection principle. What does that mean? If you look in the analytical space, you're going to see dozens of different detectors. You have mass spectrometry, many different types of mass spec. You have UV detectors. You have flame ionization detectors. MRR can be integrated into a lot of traditional workflows as a detected principle, adding all of that value and precision and unambiguous results along with traditional workflows. And so the more that we unlock, the easier we make it to integrate MRR into a traditional workflow. We are going to continue to grow that Venn diagram of applications that MRR can do too, and give you that orthogonal technique.
Currently, are you able to integrate across platforms, particular partners that you are working with, or is this something that is now on our sights for the future?
We started out with the basics. So we integrate into a traditional kind of GC workflow. We have a auto sampler that allows us to directly inject liquid samples into the instrument or interface with aerosolized or gas phase samples right off the bat. And then we also have a headspace auto sampler. We are evaluating additional techniques of sample introduction into the instrument, and we're always interested in identifying opportunities to collaborate with folks that have experience with some of their traditional techniques. And we are more than welcome to partner and try out new techniques to integrate into MRR.
So one final question I like to ask is, if you could change anything about the biotech tools and services industry, whether that's how they do funding, the procurement process, maybe the adoption of new technologies in the space, would you? And if so, what would that be and why?
That's a great question. If I could dream for a moment, I mean, you hit on a few of them that would be really valuable. I mean, of course, if we could improve funding, that would make everybody's objectives easier. But if I could pick one thing kind of in my role, focusing on the commercials, I would say it's the procurement bias towards familiar tools and legacy workflows. Technology is moving so fast, and the needs of our industry require so much more data to be integrated into our workflows that I'd love for us to move towards more of a rewarding kind of outcome-based thinking. We could establish new techniques, new applications, new workflows much faster than we do if we broke that paradigm of just depending upon that golden standard rather than trying new things. If the data is coming better, faster, more clearer, then we should be faster to try it, compare it, benchmark it, and adopt.
We'd like to thank our guest, Damien Fischer, Chief Commercial Officer at BrightSpec, for joining us on the Biotech Stack. For more information on Damien or BrightSpec, please check the link in our show notes below. Thanks for listening, and we'll see you next time.